Good morning, folks. It's morning about 11:11, and so what I have been asked to do is to make the video and then put it into D2L, so you don't have to be there at 8 o'clock on Tuesday morning or 10 o'clock or 1 o'clock or whatever. So what we're going to do today is we're going to look in Chapter 2 on chemistry. I'm going to go back over lipids a little bit, and we're going to see if we can get to the end of this chapter with the nucleic acids. Good bit of information in here. It's really fascinating to me, and I hope it will be to you also. But let's look on page 52. And on 52, we're in the second column about lipids. So as you look down page 52, second column, and you see the heading lipids, you see it says like carbohydrates in that paragraph, lipids include fats and oils, and they contain only carbon, hydrogen, and oxygen. Now, if you look at the carbohydrates and you look at the lipids in terms of the number of atoms of oxygen in the two groups of organic compounds, you're going to find out that the lipids do not have nearly as much oxygen. Got a lot of carbon-hydrogen bonds in the lipids. And then you have this group. If you come about halfway down the little paragraph, you see C15H31 COOH. That COOH, that's where the oxygen is. You don't have as much of it in lipids as you do in carbohydrates. If you, let's see, it's called a carboxylic acid. Hang on one second. Okay. Great, says my connection is unstable. But if you come down in that column and you look at fatty acids and triglycerides, you will see the third line, a fatty acid consists of a carboxylic acid group. Of course, if it's an acid group, that means it can release a hydrogen, doesn't it? That's what acids do. They can release hydrogens. So you find that carboxylic acid group in these lipids. Now, still go back into that first paragraph and look at the last 4 lines of that paragraph. You'll see it says lipids will dissolve in and can act as a solvent for other non-polar compounds. There's no polarity in those lipids. That's why they call them non-polar compounds. And so you see they often, in the third line down there, it says which are often said to be lipid soluble, those last 4 lines. So what we want to do is remember that those lipids are non-polar. Now, having said that, we've already talked about the carboxylic acid group on a fatty acid. And if you look to your right, you look on page 53, and you can look up at the top and you'll see that a fatty acid can contain anywhere from 4 to 20 carbon atoms. They can have single bonds between the carbon molecules. They can have double bonds between the molecules of carbon. So as you come down in that first column, you see that saturated fatty acids. When you think of saturated fatty acids, you want to think of single covalent bonds between the carbon atoms. Not only that, but you want to think of them as solids. I think all of you have probably browned ground beef at one time or another, or cooked bacon. And some of you may pour off when you cook it, you pour off the oils that are there. And if you let them sit for a while, you will have a layer of solid fat. So that's what they call a saturated fatty acid. It's solid, and as you come down to the heading saturated fatty acid, and you come down to the little illustration where you see the carboxyl group and the hydrocarbon chain, there are all single bonds between the carbon atoms, which puts it in that category of being saturated. Now, those are the fats. Although they're good, we all enjoy them, especially bacon. You know, you can put bacon on everything. Never tried it on ice cream, but it sure goes good in sandwiches and various kinds of salads and so forth. So if you eat too much of this, it can clog your arteries. It's better to limit the saturated fats. I wouldn't say just get rid of them, but you just need to not take in as many as maybe you would like to take in. It's more of a healthy lifestyle to avoid, to a degree, the saturated fatty acids. You don't want to go way out into Never Never Land in terms of what you eat, but a blend of things. But remembering that the saturated fatty acids can destroy your cardiovascular health. So you see, that's what it looks like, a long straight chain of carbon atoms. And so they label that one, that saturated fatty acid, as palmitic acid. You don't have to know that name. Just know that when you think saturated fatty acid, you think of solids, and you think of carbon atoms having a single covalent bond between carbon atoms. You come down to the next two, and you will see, as you look at B and C, they bend at that particular point where a double covalent bond is located. So you see monounsaturated fatty acids, and you see down at the bottom polyunsaturated fatty acids. So if you got two or more double bonds in the fatty acid, they call it polyunsaturated. Now, those are good oils. You get them from salmon, you get them from other fish, you can get them from sardines, you can get them from nuts, peanuts, almonds, walnuts, and things like that. Those are good fatty acids. They do not clog up your arteries as the saturated fatty acids have a tendency to do. So again, there you want to eat some of those good things like that. Olive oil, there are olive oil blends that have that content of monounsaturated and polyunsaturated, just like peanut butter. My father was a cardiologist, and he didn't really think very highly of me liking peanut butter. Of course, in grad school and other places where as I moved along in life, peanut butter sandwiches and jelly and bananas, that's what sometimes you had to eat because you didn't make a lot of money. But then they found out that these peanut oils are good for you. My father thought all those oils might be clogging you up, but he only knew a certain amount. So, we don't have a handle on everything. We are growing in our understanding of various aspects of life. So you will see that happen in your lifetime too. But anyway, you want to eat some nuts. Those are good. They go great, as peanut butter. They good with salads and so forth, and other dishes. And then of course fish. You hear about the omega-3 fatty acids. Those come from those fish that we mentioned. So that's a good diet. And of course, they want you to do some exercise too. So it's a balance. It comes back to homeostasis, a balance of things. You don't want to go out too far on a limb. We want to stay sort of in the middle of the road. Get some exercise. Watch your diet. Don't overeat. Don't eat all the saturated fats you can get. So anyway, you got the picture there about the unsaturated, polyunsaturated, monounsaturated molecules and the saturated ones. Now, When we eat those oils, what our body does sometimes when we have enough of them, maybe a little bit more than we really need to use, is we will store them, but we don't store them as fatty acids. On page 53, you come about halfway down and you see in the paragraph, it says our bodies cannot package fatty acids into long chains as you can do in terms of polysaccharides. So what we have to do is you look in there, you see that bold print term glycerol. That's a 3-carbon alcohol. And we can combine fatty acids with glycerol, and we get what is called a triglyceride. And the triglyceride can be stored in fat cells. As you see down at the bottom half of that column, here we come up with some of the chemistry that we talked about in terms of reactions. You see the glycerol to the left, we see the fatty acids to the right, and you notice in blue on the fatty acids, you see the OH, and you look over on the glycerol, and you see the H is blue, has a little blue bubble around it. There's an OH and an H, and they're going to be put together to produce water. Look at the arrow coming down, and you see the term dehydration synthesis. This stuff is going to come back. You know, we covered it back in the other early in chapter 2, and you're going to be hearing it all during the semester. So you don't forget what is in chapter 2. It's going to be a foundational chapter. We'll keep bringing things up to you. The more we bring them up to you and you start saying, gosh, they just play a role in all kinds of aspects of our body. So notice it says dehydration synthesis. Is that an anabolic reaction or is it a catabolic reaction? Answer the question. Think about it. Somebody might ask you something like that, like me. And you see those water molecules are pulled out of there. And now you have down at the and you look in the first column and you see it says that you come down to the second paragraph. That's where I'm interested that you look at. And you see it says triglycerides are stored in fat cells. You know, some people go crazy about, their weight or whatever. Homeostasis, okay? Not too far to the right, not too far to the left. We have to have fat. We have to have triglycerides. You see it's stored. Most of us think of it being stored around our waist or some other places. We don't want it to be too much. And you see it says, as you read that, it helps to insulate our internal organs. Now, one of the things it does is it supports your kidneys. You don't want to lose that fat in your abdominopelvic cavity. It not only supports your kidneys, but it protects them. It surrounds them. So if you get hit back there, hopefully the fat takes up more of the blow than the kidney does. So it supports organs. You see, it says it protects against physical shock. You want some fat around your joints and so forth. It's like a cushion. It's like a cushion. Some of those big guys that play in those real tough sports like football, they've got some fat on them, and that fat gives them some protection. Not only keeps them warm, keeps the organs inside warm, but it gives them a cushion. And then you see a second thing, or third thing is that they can regulate your body temperature. And if you can go on down to the third sentence, triglycerides are a supply of fuel. We can burn the fat. So it's good to have some. Even physicians have said that it's good to have a little fat around you. You don't want to go too far either way. Remember, homeostasis. All right, that takes care of the lipids. Now let's mention a little bit about phospholipids. Here we're going to have a lipid, but it's going to have some aspect of phosphorus attached to it. So as you look in the second column, you see the heading says phospholipids. And as you read down, you'll see it'll tell you in terms of a triglyceride, you're going to have, in place of a third fatty acid, you find a phosphate group. Got phosphorus in there, got nitrogen in there, and it's a very important molecule. Now look at the little picture to the right. It says schematic structure. You're going to see a lot of that, especially in chapter 3. That's a little schematic drawing representing a phospholipid. And one of the things that you may know, if you took 110, you may remember this, that the cells in our body are made up of a phospholipid bilayer. You can't get away from lipids. You have to have lipids. You just want to balance it off. And sometimes it's hard to do that. You know, Thanksgiving and Christmas come around, and we start thinking, oh, man, we're going to eat. I have this same struggle. It's a choice, okay? Balance it out. Enjoy it. Sometimes we have to cut out a little bit here because we've eaten a lot more over there. But it's a lot of fun, isn't it? Just a wonderful time with family and good food. But you look at the little schematic, and you're going to be seeing that over and over and over again. You look to the left of the schematic, and you see this big picture. It's figure 219. And you see it is showing you all the molecules involved in a phospholipid. So as you look up at the top, you see coming down the center of there, you see nitrogen, you see carbon, you see oxygen, you see phosphorus. And that portion of a phospholipid, go to the right, and you see it says it's hydrophilic. Remember that term? Likes water. So substances that can dissolve in water, like that head, so to speak. You come down into those two tails that are hanging down. They're called fatty acids. Those are non-polar. They are hydrophobic. So the top portion of a phospholipid is polar. The bottom half of it, just past the glycerol in the middle, you see that it's a non-polar situation. That's going to be developed as to how that helps us in terms of having that as a cell membrane. Real interesting stuff. It's amazing. Works very well. All right, that's all we're going to say about phospholipids at this point. You know they play a role in the plasma membrane. Now we move over. Oh, there's one more thing. Excuse me, I was about to leave this out. As you go to the first column on page 55, and you come down about 1, 2, 3, 4, 5 lines, you will see in italics the word amphiphilic. You probably have heard of amphibians. Amphibians can live on land and water. Okay, they can go both ways. So amphiphilic means that it likes both aspects. Sounds like somebody ought to run for office, right? Talk out both sides of their mouth or something like that. Not that they all do that, but we have that tendency to do it as people. But anyway, there's a section that's nonpolar. There's a section that is polar. Amphiphilic. Amphibian. Land, water. Okay. So that's why they call it an amphiphilic molecule. It has the polarity in one part of the molecule, it has non-polarity in another part of the molecule. Now, let's leave the phospholipids and go to steroids. And you see it says the class of lipids that share a 4-ring hydrocarbon structure. Look at the top of page 55, and you see what they call the steroid nucleus. And then you see below that, that's steroid nucleus. But you see all these functional groups. They call these, where you see the CH3 and the CH2 and so forth, and the OH, and they're added to a molecule. They call them functional groups, meaning that they create a particular function in the body. They can be very similar in terms of the 4 ring structures, 3 hexagons and 1 pentagon. But you start adding these functional groups, and we come up with a molecule that carries out a particular function. Now, I want you to come down where you see steroids. Come down to where you see steroids on page 55, and come down 1, 2, 3, and start with line 3. The steroid cholesterol forms the basis for the body's other steroids. You know, we have maligned cholesterol. That means we've said bad things about it. We got to have cholesterol. Cholesterol is necessary, and we'll see how that works out in just a second in this, but it's also a part of our cell membrane. We need to have it. Some people try to eliminate it. Oh, and then I'll just live forever, you know, or live for 80 or 90 years. You got to have cholesterol. Homeostasis, the balance again. That's why I started this class off with homeostasis. It's critical to have a handle on homeostasis and see what we can do to keeping things balanced. Anyway, as you come to that third line, the steroid cholesterol forms the basis for other steroids. Now, we don't get into bile acids right now, but notice it says the sex hormones, estrogen and testosterone. Estrogen, your girls got to have it. It's involved in the cycle of your ovaries and your uterine wall and so forth. And then testosterone is important for the guys. Yeah, girls produce a little testosterone, so do guys produce a little estrogen. But they have their roles. But they all have this same 4-ring structure. But you put different functional groups on there, and it does something different. It's pretty neat, isn't it? One of the things that you guys will run into is you will, and you may have some people in your family that are doing this, that are taking a drug called prednisone. Sometimes they call it prednisolone. That's the basic formula. It's a steroid that reduces inflammation, has side effects. That's why you don't want to take much of it. You want to take a little of it, as much as is needed to maybe remedy a situation. But it tends to reduce inflammation, and it's been good for people, but you can take too much of it. So you can read about that, and some of you may know about that and some of the side effects that your family members have endured. Okay, so much for steroids. You think of cholesterol, but also think of estrogen, think of testosterone, think of prednisone, all part of life. Again, balance, the right amount. You know, there are people who try to take testosterone, get all those big muscles and so forth. You don't want to take too much of it. You can actually take too much water. Do you know that? You could actually take too much water in your system, and it can be damaging to you, dangerous. Homeostasis. Now, let's leave that. Don't forget to look at the quick check at the top of page 55. Let's go into proteins now. We all talk about eating protein. You get protein in cheese, you get protein in beef, you get it in fish, and so forth. We need protein. And as you look in the column on 55, you see in the second sentence down over to the right, protein makes up your hair. I don't have to have as much protein now as I used to because I had a head full of hair a long time ago. Makes up your nails, part of our skin. We'll get to that a little bit in chapter 4, I think it is. And they're needed in our bones, as you see. And you see we produce anywhere from 50 to 100,000 different proteins. Now, I'll preface the next section with this thought, thinking about that. For every protein, there is a gene in our body. 50,000 different types of proteins. So there's a little bit of contradiction in some of the stuff that they say now, because they say, well, we have about 25,000 genes. Well, that doesn't jive with this, does it? So, science does not have everything figured out. Don't think it does. You're going to learn enough to where you can be a blessing to people and a help to the community through your profession. But we don't have all the answers. But that's part of the fun. It's fun to delve into that and see what people have done and how we've discovered various things. And then sometimes it's helpful. So anyway, as you come down to in that paragraph, look on the left-hand side as you come down that paragraph with proteins as its heading, you see the bold print protein, macromolecules. They're big molecules, relatively speaking. They can act as enzymes. They can, let's see, where's it? I got something in my eye. They can protect you. You've heard about the, coronavirus, COVID-19. People who have made it through that disease, says defenses. What are they doing? They're giving people the serum, and sometimes a direct blood transfusion if their blood types are compatible. But in their blood, they've got these defenses, proteins that will attack the virus. And people have taken that from other people, and they have recovered several days earlier. So we've got proteins in our immune system that we'll talk about maybe in 211. Anyway, you work your way through there. You see it talks about muscles contracting and so forth. The muscles that are running my mouth right now, the muscles that are handling my intake of air and my respiration, my exhalation of air, the muscle of the heart that's beating for me, protein. We take protein in like turkey. We break it apart and then we reassemble it to fit our proteins. Anabolism, catabolism, okay, keep that straight. Dehydration, synthesis, hydrolysis. See, this stuff all comes back into a very useful part of the program, of the course you're taking. Now, come down to where you see amino acids. Those are the monomers of proteins. There are about 20 different amino acids that are arranged in a particular sequence, particular number, okay, and a particular type of amino acid. We've got 20 different amino acids. How we arrange them determines what the function of that protein is going to be. It's like the letters in our alphabet. We learned letters, and we arrange those letters into words on a paper, like you got on your Word document, and we know what those words mean. As you read your Word document I gave you, maybe over there in nucleic acids, you change the letters on words and they don't mean the same thing. Like step. You think step, Your feet are moving, or you think of these little treads that you climb up to go upstairs to your bedroom or something like that. But if you change the E to an O, it doesn't mean that. Different word, different function. Same thing with amino acids. How they're arranged, what number there are, and what amino acids are used makes for the function of a particular protein. Proteins are very specific in their duties, their functions within the body. Now, coming down into the amino acids, I want you to look over to the left at the bottom, figure 2121, and you see the illustration of an amino acid. They basically have three things, three components, and as you look at the left, you see the little block down there that has the N and the two H's. That's called the amino group. It's got nitrogen in it. We have to have nitrogen. Now, don't go eat any fertilizer that you put on your yard or on your plants. That's not what we're talking about. You got to get it from other proteins, okay? Like your turkey that you eat. You see, there's your amino group. And here's that central carbon just to the right of it. Every amino acid has a central carbon. And then you see an H down below, and then you see a carboxylic acid to the right. It's an acid, amino acid. That means it can give off a hydrogen. And then you see the R group. And notice what it says. It determines the amino acid's properties. In other words, what is it going to do? Okay? They give you some examples of amino acids. Do not go and memorize them. I had to do that in grad school when I took an intermediary metabolism and biochemistry course. It was a lot of fun. A lot of memory. Good grief, there's a lot of memory about all these different amino acids and hooking together and getting all these particular compounds. It was a lot of fun, but it was a challenge. So now you know the three components, the three basic components: amino group, central carbon, carboxylic acid group, and then the R group, which can be variable because 50,000 different proteins, 100,000 different proteins. They arrange the 20 amino acids. I say they, our DNA and our RNA, which we haven't talked about yet, that does that for us. It allows me the freedom now to sit here in front of this camera and talk to you and tell you about that kind of stuff. And when you're taking a break and you're watching Jeopardy or something like that, you're breaking down those proteins that you ate. Maybe it's a grilled cheese or something, and you're taking that cow's proteins that produce that milk that we turned into cheese, and you're breaking it down, turning it in from cow proteins into your protein. That's what we do. We take that and we turn it into us in the body. Isn't that amazing? And you don't have to worry about that at all. You just enjoy Jeopardy. Your body is programmed, engineered, designed to do that. So you're free to do some fun things, right? Like take this course. Okay, so now you know about amino acids. Let's look over on page 56. And I want you to look at the bottom of the first column, and you see peptides and peptide bonds. So you see in the example down below, you got two amino acids. Don't go know their names. We're not going to get into that. They just list them, alanine and glycine, fairly simple. But you see they have the same three components. Got an amino group, got a central carbon, and got a carboxylic acid group. And then notice they are different in their R group. They would call that radical group. CH3, yep, CH3, it's a pretty small 3 for me to look at. And then an H. But that helps determine its function, see? So you see those two amino acids, got a plus between them, we're going to have a chemical reaction. And you see going to the right, follow the top arrow, dehydration synthesis. What kind of chemical reaction is that? Okay, remember catabolism and anabolism? Okay. Here goes the water, and now they're joined, you see, by a peptide bond. It's a bond between the N, the nitrogen, and the carbon that's got a double bonded oxygen on it. That's not your central carbon. That's to the next one, a little bit to the left. But that's part of the carboxylic acid group. So that's dehydration synthesis that begins to make a peptide bond. And you see they call it a dipeptide. Look right below that. Two amino acids joined together. If you add another one, you have a tripeptide. And I think after that, they simply call it a polypeptide. Here comes that word back from polysaccharide. Okay. You and I, let's see, what time is it? It's about 11:48 right now. So about 30, 40 minutes, I'm going to go get some lunch. And what I'm going to eat, I'm going to break down the protein of some turkey or a chicken, or maybe the protein of beans or something like that. And I'm going to break it up. I'm not going to have to worry about it. My body's going to do it. I'm going to break it up into amino acids. As you see, hydrolysis. That's one reason you need water in your system so that digestion can be efficient. And you break those bonds, you break the peptide bonds, and you get individual amino acids. And your body reassembles those into the proteins that are needed for our skin, your hair, your fingernails, your muscles, your bones. We just enjoy eating, don't we? We trust the food to be non-toxic and healthy for our bodies. So, come over to the second column, and right at the top, very top, you see polypeptides in bold print. Okay, that's a bunch of them. Could be anywhere from like 5 or 6 or 10, or could be 150 of them or whatever. But look, it says contain 10 or more. Okay, so you see proteins over to the right, bold print. Notice what it says. Consists of one or more polypeptide chains that are folded into a specific structure. They form a specific physical structure. That physical structure will determine the function of that protein. Will it be protein for your muscle? Will it be protein for an enzyme that breaks down food for you? Depends on the shape. If you change the shape, you change the function. Okay. Got to let that register in your head. So when you see protein structure, come down and you see the 4 levels of protein structure. Primary, that's the chain. Now, if I were to ask you it's okay to say it's a chain of amino acids because it is. That's how it starts out. And it still is a chain of amino acids. The protein that we use in our fingernails is different from the protein that's in our stomach, digesting our lunch. The type of amino acid, the sequence of the amino acid, and the number of the amino acids. Those 3 things make the difference. what amino acids are used, what sequence they're put into. Makes all the difference in the world. And you get a particular shape. If you change the shape, it's not going to do what it's supposed to do. And there are situations in our body where the shape can be changed and we can have some big problems. We can even go into eternity if we change the shape of the proteins. But anyway, come back to the bottom of that second column on page 56. Second column, you see the primary structure. That's a sequence of amino acids. Somebody's calling me. Oh, can you hang on just a second? Hello? Hi. I think she's downtown getting some groceries and things like that. I appreciate that. I'll tell her you called. Okay, bye-bye. Neighbor calling us. Even I have a problem sometimes with the phone. I'm trying to do this just like you guys in terms of you'd come to the lecture. We try to get you to cut off your phone. Anyway, you can see the secondaries. For the primary structure is shown up top. You can see it's a chain of amino acids. Secondary structure, you can see they call it beta pleated sheets or alpha helix. That's part of the shape that is going to make that protein do a certain job. And then you have tertiary structure where the R groups, remember those R groups? They can be all kinds of things on there, but they determine the shape of the protein after it's folded. They help determine the shape of the protein as soon as after it is folded. And you can have globular, so you got quaternary down there too, assembly of two or more chains of amino acids. And they all intertwine in a certain particular form to carry out that function. And so you see you have sort of globular proteins. Those would be maybe enzymes or something like that. But then look over here, you have these fibrous proteins that look like cords maybe wrapped around each other. And we'll talk about those when we get into chapter 4 on tissues, because we have proteins in our skin called keratin, in our nails, tough little protein. And then collagen's another one that strengthens tissues like our bones and so forth. And I think it's even in your hair. So those are the 4 levels of structure of a protein. So as you read through primary, secondary, tertiary, and quaternary, develop a definition. Quaternary structure would be maybe 2, 3, 4 proteins intertwined with each other. That makes up one protein. Polypeptide chains, all in a particular fashion. It's not by chance. It's done by code, by instructions that we'll talk about in just a few minutes. We mentioned that proteins could be broken up. So you look down the second of the first paragraph, you come down to the section where it says protein denaturation, and that's where the protein comes apart. Maybe due to, well, as a matter of fact, look under denaturation, the bold print term, and read down to the end of that paragraph. It's going to talk about temperature. That's just one thing. But you get a fever. If it goes too high, the proteins start to unravel and you can die. Excess temperature. Okay, so much for proteins. Come down, you make sure you do the quick check. Now we're going to look at nucleotides and nucleic acids. So you're up at the top of the second column, and you see it says the last class of organic molecules is nucleotides. Nucleotides, as it's going to tell you, are monomers. You know, we had amino acids that were monomers. We had monosaccharides that were monomers. Right? Excuse me. And now we have nucleotides. They're the monomers that form our genetic material. We got two kinds of nucleic acids, DNA and RNA. You probably already know about that. And they are extremely valuable, very important in our livelihood. So let's come down to nucleotide structure. You know they're monomers. They're the repeating units. And you see there it says each nucleotide has three parts: a nitrogenous base, a 5-carbon sugar, pentose, and a phosphate group. You look at that below. Everybody see that? Figure 2.24. That hexagon represents a nitrogenous base. You can see the pentagon represents the pentose sugar. And then you got the phosphate group. That's the acid part of the compound too. That's why they call them nucleic acids. Okay. And so you can change the type of nitrogenous base, and you can change the type of sugar. Well, we got two kinds of sugars now here involved in this: ribose and deoxyribose. Go back to the carbohydrates and look at that across the top of the page where you see galactose and glucose and fructose. Then you see ribose and deoxyribose. We don't break down deoxyribose and ribose. We probably don't have an enzyme for that, but we don't break it down. We simply turn it back into us. Every time you eat broccoli, you eat these nucleotides, and you rearrange them so they're not going to produce broccoli coming out your ear. They're going to be involved in produce the cerumen, the wax that produces that's in your ear. Some cell is going to have that capability to produce that. Your skin doesn't do that, but this does. Kind of interesting, isn't it? Doesn't do that on here. Doesn't do that under here. Does it in your ear? How do they know to do that? We don't have all the answers. That's part of the fun of it. We just sit back and go, great, now look at that. Now, let's break it apart a little bit. As you look at the nitrogenous base, you see that arrow coming down. You got two blocks. One block they entitle purines. One block they entitle pyrimidines. You see you got a pentagon and a hexagon for either one of those two substances, adenine and guanine. You see they got nitrogen in them. A good bit of nitrogen in there, aren't they? You look at the ends. Got double bonds between the carbons, which we've talked about before. So those are the two purines. They are nitrogenous bases. And then you look at the pyrimidines, and you got three of them. And I think it's kind of easy to remember that they're the pyrimidines. Number one, let's think about this. The purines, up there where it says purines, they have a smaller word, don't they? They have 1, 2, 3, 4, 5, 6, 7 letters for purines. Two purines. Come down to pyrimidines, which has more letters, and you have three other nitrogenous bases: cythymine and uracil. If you rearrange that, you can remember CUT, C-U-T, cytosine, uracil, and thymine. Now, you guys probably you've already been involved in cuts, right? You've been cut before. If you're going to get into the medical profession, you might be getting into a lot of cutting. Okay. So think of CUT, cytosine, uracil, and thymine. And since there's three of them, You got more of them than in the pyrimidines, which has more letters. And then you have purines, which has smaller number of letters, has only 2 nitrogenous bases. So those are the 5 bases. And then you have the pentose. And as I said, you can only have deoxyribose or ribose. So as you look at that diagram on page 224, when you have the pentose sugar ribose, when you've got ribose in there, then the nitrogenous bases, this is something you just have to memorize, the bases that would fit over there in that hexagon, the hexagon is just an example, okay? It's not the actual whole structure of the nitrogenous base. But if you got a ribose sugar, then you're going to use uracil. You can use uracil as a nitrogenous base, guanine, cytosine, and adenine. Those 4. Ribose does not use thymine. Okay? If those would be called ribonucleotides. If you have the sugar, the pentose deoxyribose, then you don't use uracil. DNA does not use uracil. Deoxyribonucleic acid does not use uracil. It uses thymine, adenine, cytosine, and guanine. You got to remember that. Uracil only belongs to ribonucleic acids. So the deoxyribonucleotides are the ones that make up DNA. Deoxyribonucleic acid. And then RNA is made up of ribonucleotides in chains. Let's look. We're going to come back to ATP in just a minute, but let's look over on 59 and look at nucleic acids and DNA. Everybody see that? Page 59, nucleic acid, DNA, nucleic acids, DNA. Now look at, this is real important here. Nucleic acids account for only 2%. That's not a whole lot, is it? But boy, is it important. There is more information in that nucleus than there is in 100 sets of the Encyclopedia Britannica. You ever heard of that? That's a big encyclopedia. Got a lot of information in there. That's what DNA is. It's information. Intelligent information. As you're looking on page 59 and you come down to the nucleic acids, DNA and RNA, only 2%, but they are some of the most important molecules in the body. You monkey with them and you can end up with a mess. There are things that are genetically transmitted, aren't they? We take in sometimes some chemicals that can change the DNA. It's information. If you change the information, you change structures, you change abilities, you change functions. It's fascinatingly intriguing. It really is. It's information. Come down to the third line, the 2 main nucleic acids are deoxyribonucleic acid, or DNA, and ribonucleic acid, RNA. Now notice, next sentence, together they are responsible for the storage, just think about that, the storage of the genetic code. We talk about our genes that are in the nucleus. We'll learn about that, when we get to chapter 3. And there are molecules that actually go along the DNA and check it out to make sure it's right. Most of the time it works great, but sometimes something happens. And then we have this problem. Just blows my mind, but I love it. I hope you do too. And now it says the execution of the genetic code. Now we think of an execution, we think of taking somebody's life, don't we? Or sometimes you might think, well, let's execute this program, which means to carry it out. So when they talk about the execution of the genetic code, they're really talking about the expression of the genetic code. That's why I look different from you. That's why I have these little lobes here. Some of y'all don't have those lobes because your DNA is different. And you could go on with all the features. You know, we all have the same body plan, don't we? But we look at each other's faces and we recognize each other by the features. Those features were made through DNA. So we're all different. Have different blood types. Some of us got the same blood type. Just fascinating stuff. Let's go over to page 60, 61. Those are the last two pages we cover. All right, you know about nucleotides. You know that the nucleotides hook together into a polymer, okay? The nucleic acid. Nucleic acid is a polymer. Nucleotides are the repeating units or the monomers, okay? So you look up at the top and it says nucleic acids are polymers of linked, connected nucleotides. And look what it says. Here we come back to those chemistry things, you know. The nucleotides in a nucleic acid are linked by dehydration synthesis. It ain't going away, folks. Get it down. You're going to see it. Always review this stuff. And you see it says it joins the phosphate group of one nucleotide to the sugar of the next by a polar covalent bond. Polar covalent. These would tend to dissolve in water because they're polar covalent. But you see what's hooked together. Now, let's do this. Let's go to the middle of the page. You're looking at figure 226, page 60. Look in the middle of the page and look to the left, and you see it looks like a purple ribbon that is just a couple of purple ribbons that are twisted into a helix. And then you see these little colorful steps in between, bars, if you want to use that kind of word. Now, look down at the bottom for just a second. Look at the bottom and over to the right. We're on page 60. And you see it says key, adenine, guanine, thymine, cytosine, uracil. Got different colors. Okay. Now remember, uracil is only found in an RNA nucleotide. It's not found in any DNA nucleotides. And the thymine nucleotide is found in DNA. It's not found in RNA. Now, we can get into why that is if you wanted to, but I'm not going to get into it right now. So when you look at the coiled ribbons, if you want to think of that, you can. A lot of people would untwist this, and you would have what looks like a ladder. The ladder has rails, and it has steps. So some people say it's a twisted ladder. That's okay if that helps you understand it. Nothing wrong with that. They just use the word double helix quite often. And so, but now you understand what they're talking about. Now notice we're back in the middle of the picture, and you see in the middle there of the double helix, which is DNA. DNA is always double helix, except in viruses, and then they throw us for a loop. But bacteria have double helix DNA. You and I do. Horses do. Cats do. Look what it's pointing there in the middle. It says sugar phosphate backbone. It's pointing to the ribbon. kind of purple ribbon or purple rails, okay? Sugar, phosphate, backbone. Sugar, what sugar's there? Deoxyribose. And then a phosphate. Let's take what you're looking at there in terms of that purple ribbon, go right up to the left, and you see a picture. It says bases, sugar, phosphate, backbone. And where they have that purple box around that pentose and the phosphorus, that's the rail like a ladder, okay? And you see it's a sugar, phosphate, sugar, phosphate. That's the rail. So if this is DNA, when you see that pentose is going to be deoxyribose, and then there's going to be a phosphate group that connects it to the next deoxyribose. And you can get into hundreds and thousands of those in the polymer DNA. Okay? So you see what the rails are, and then you see, if you look out to the left, you see the little hexagon that they use as just a standard presentation of a nitrogenous base. See, that's a nucleotide. That whole thing's a nucleotide. Nitrogenous base, the pentose, in this case, deoxyribose, and then the phosphate group. That whole little section, those three components form a nucleotide, which is the monomer of DNA. DNA is a polymer of nucleotides. Now, as you look, come back down into the middle where you see the double helix, and notice the little nitrogenous bases represented by those colorful little blocks, and then they got A in there and T in there. Look through DNA. You see T combines with A. You want to know this. C combines with G. There's another A and a T. There's a C and a G. There's an A and a T, a C and a G and a C. As you work your way down, they always combine that way. Now, the sequence of those nucleotides that you're looking at in DNA, a sequence of those is a gene that determines that this doesn't have, that my lobe is loose, as opposed to yours not being loose. That's why we're different. That's why we can identify each other. So you don't see uracil in there, do you? DNA does not carry uracil. But notice that adenine always combines with thymine, guanine always combines with cytosine, or you can reverse it and say thymine always attaches to adenine and cytosine attaches to guanine. See the little dots between the the nitrogenous bases? Look what it says they are. Do you remember that? Hydrogen bonds. That's one reason we say this is a polar molecule. Remember that in that water, you got positives over there on the hydrogen, negatives on the oxygen. And we have hydrogen bonds. It makes the water a little denser. That's why you can float a coin across the water if you can put it on there carefully enough. So DNA, we have 23 pairs of chromosomes that are built like this. Those carry all the information for the formation of this body. You don't see somebody's arm coming out their buttock. You don't see toes hanging off their ears. That's because there are genes that direct the protein production, which directs the construction of our body. We do not understand all that. But boy, it's fun to look at and learn about it. It's amazing, just amazing. All you got to do is eat, sleep, and go to the bathroom. Everything works. Just flat out fascinating. So DNA contains the code. RNA to the right is always single-stranded. And you see it has a sugar phosphate backbone, but instead of having a deoxyribose sugar, it has the ribose sugar. Sugar linked to another sugar by a phosphate, and that goes all the way down. And then you see the nitrogenous bases, but you notice what's missing in there in terms of nitrogenous bases. What is that? What's missing? Thymine. RNA does not carry thymine. It carries uracil. We don't necessarily understand all that, but we work with it. You don't understand the transmission in your car. I don't either. But you don't have a problem putting it in R&D so you can get where you need to get. So sometimes we know enough to make use of something, but how it works, we do not understand always. Oh, there's where I wanted to write that down. Yeah, nitrogenous base. So adenine pairs up with thymine in terms of DNA. Guanine pairs up with cytosine. We're not going to get into why. It has to do with the hydrogen bonds and so forth. But DNA has the information that we need. This RNA molecule, always single-stranded, except in viruses, and boy, they throw us for a loop, as they're doing now with the coronavirus. But the RNA is always single-stranded, and amazing dance. Just amazing. But we don't think anything about it. We don't appreciate what happens until it goes bad, until something happens, maybe taking a poison or something like that. I want you to, as you look on page 61, you see these little bullets here. DNA contains the sugar, deoxyribose. DNA contains the adenine. We've already talked about that, but you want to know them. The two strands are held together by hydrogen bonds and so forth. And then DNA exhibits complementary base pairing. What in the blazes does that mean? Come down to the third line. However, a pattern is followed throughout the entire molecule. The purine adenine always bonds with the pyrimidine thymine. That's in DNA. Okay? Keep it in DNA at this point. And the purine guanine always bonds with the pyrimidine cytosine. That is base pairing, complementary base pairing. They fit each other right in terms of bonds. Now look at the second column. And you see within the DNA, we find the genetic code. All that information for the formation of this body and the maintenance of this body. Now, I told you not to eat too much of this or too much of that and get some exercise and things like that. Those are all good because we can exceed the control of things in our body if we're not homeostatically minded. It's got the code in the DNA. You see it says it's a recipe for each and every protein in our body. This is on the second column, page 61. The recipe for one individual protein is found within a segment of DNA, and that segment is called a gene. Now, I've heard some new information that I don't understand totally, but I sure do appreciate it. I'm not going to get into it, but you see it says after that gene, the sequence of the nucleotides within each gene ultimately specifies the amino acid sequence of a protein, a specific protein that's going to carry out a specific job. Not going to do something else. That's why we need 50 to 100,000 proteins. And you see it says the cell can read this sequence. The cell reads. Yeah, we use those kind of words. And assemble the encoded protein via a process called protein synthesis. It's going on right now in your body. It's going on right now in my body. And I'm getting a little hungry. It's about 12:23. But it's going to go on. I got to make skin. I got to make hormones. I got to make proteins that guide the metabolism that's going on in my body. Come on down to the next paragraph. Protein synthesis requires more than just DNA. DNA is the code. In fact, DNA is not directly involved in the protein assembly. It's got the code, but the RNAs play the role in assembling the amino acids that we need for our health. The task of assembling the correct amino acid for a protein falls to RNA. Now, we're going to get that at the end of chapter 3. I'm just about through with making a Word document with that, and then I'll move into chapter 4, which is dealing with tissues, where we look at all these different tissues that make up our bodies. But you see it says, still reading there, unlike DNA, RNA consists of only a single strand, okay, and it is not confined to the nucleus. In addition, RNA contains a sugar, ribose, and the nitrogenous base uracil. Now, we've already built that foundation for you there. This is just repeating what we covered. Uracil binds to adenine in the same manner as thymine, so they physically fit together. Then uracil, it says, and when an RNA molecule binds to another nucleotide strand, U pairs with A and C with G. Look at the next paragraph. Crucial role in protein synthesis. So the code is in DNA, but RNA carries the code out of the nucleus. We'll see how that's done a little bit later, but it carries the code out of the nucleus, joins to some organelles that some of you may know about. They're called ribosomes. We'll talk about them later. And they start making copies of that protein. I love this stuff. It is just, it's glorious. Now, in closing this out here in terms of the content, you see the last sentence, the two processes called transcription and translation are carried out with the help of RNA molecules. Transcription and translation. We'll get into that. Now, you should have a Word document that I ask you to review. On page 64, 65 to some degree, some of them I had them checked off, I think, and then I checked off some on page 66, it says level 1, true, false type statements. Get to thinking about that. What are number 6 on page 66? What are the hydrogen bonds and how do they form and so forth? I've already got that at the end of the document I sent to you. So I'm going to eventually put this in this afternoon into D2L so you can draw it up, because we're going to have a test on Thursday. And if you have questions about understanding something, you have my email address. And I'm certainly not opposed to sitting here and helping you guys out. I'd like to see you succeed. It's going to take work on your part. Okay? Just like it took work on my part to get here at this point. All right. Well, I'm going to conclude this, and then I will eventually put it into week number 2 of online lecture. And that'll conclude what we're going to be tested on come Thursday. Okay? I'll talk with you later.